see everybody here. Today it's my pleasure to introduce John Lick. He's the chief of the division of Hem/Onc and associate director for clinical sciences research at the Lurie Cancer Center downtown or in Northwestern. He's been the recipient of a lot of awards over the years. Leukemia Society Scholar, Burroughs Wellcome Clinical Scientist Award in Translational Research. He's a PI on many very interesting projects. We're going to hear about some of them today. He runs a Leukemia and Lymphoma Society Specialized Center for Excellence, where he's studying epigenetic mechanisms, and this is the overall theme of his own research program, but he's brought together a lot of outstanding researchers to address that in hematological malignancies in the city. And he also is a senior scientific leader of the NCI-funded Physical Science Oncology Center that I've had the privilege to act as an advisor for, and this is really an outstanding program that he's put together along with John Lidham, connecting basic and clinical science in this physical oncology center. I'm sure we'll hear a little bit of some of the research going on in that center. He's over the years been the editor of a number of different journals. Currently, if you want to know who to contact, if you want to send a paper to Clinical Cancer Research or Oncogene, he's your man. And last year he was chosen in the Illinois chapter of Leukemia and Lymphoma Society as a Researcher of the Year. And so he continues to have just a very outstanding research program. And the broad goal of this program is how to understand how mutations of transcriptional regulators may set up patterns of aberrant gene expression that are related to cancer phenotypes. And so I really look forward to hearing your presentation.
Thank you very much. Thank you very much, Kevin, for that very gracious introduction. It's always a pleasure to come down here and even see people I haven't seen for 15 years. Like Dr. Lyman's over here. It's just wonderful. So my lab, John and I started off in the Department of Molecular Biology and Developmental Biology at Mount Sinai. And I sort of used the mathematical model where I was like the odd physician up there. But we found kind of common ground. We both worked on fruit fly factors for a while. But then I had to get to my physician roots, and I've always tried to work on transcriptional regulators that, certainly may well have homologs in the fly, and most of them do, but try to understand how these transcriptional regulators may be affecting cancer, maybe causing cancer, and may be targeted for cancer therapy. So we did this for a long time on zinc-finger type transcription factors, the Kruppel-like factors, but starting in the late 90s, early zeros, I guess, we started working on another class of proteins and another disease that we worked on leukemia for a while, but now been interested in multiple myeloma, which is an increasing problem. It's a disease that's still on the rise. Like most hematological malignancies, it's relatively rare. It's non-existent in kids, really, but it's found in adults. And it's a tumor of very late B cells that's generally been incurable. Median survival has improved. It was 3 years. It's now closer to 5 or 6 years due to some very important therapeutic advances that have been entirely empirically based, not based upon mechanisms at all, but have been fortunately to be worked out. We'd like to think that if we really understood the disease and its molecular mechanisms, you might even do better than that. So this is your temperature. Let me see if I blow my hand. So if you look at the ontogeny of B cells are born in the bone marrow, and get educated in the germinal centers and lymph nodes, and then undergo further maturation to either form initially IgM-producing cells or more mature plasma cells. And of course, this maturation of B cells is accommodated by molecular genetic changes in the nature of the immunoglobulin genes, which we'll talk about in a moment. When you have a tumor of this very late B cell that's only making IgM, this is called Waldenstrom's macroglobulinemia. If it's a further matured cell that makes more mature immunoglobulins, IgG or A or E, this is multiple myeloma. So the immunoglobulin genes rearrange with variable diversity of joining segments, linking together in different orders to form a pre-B cell which has not been exposed to antigen. Upon antigen-driven stimulation of these pre-B cells, they undergo somatic hypermutation to get a very diverse set of VDJ rearranged alleles. But initially, these VDJ rearranged alleles are linked up to the transcribed and then make it immunoglobulin mu heavy chain. With further stimulation and reimmunization, you get a class switch recombination, and you loop out this bit of DNA to form a more mature immunoglobulin. Now, this is the way things normally work. You normally loop out this region. But in multiple myeloma, the molecular pathogenesis was really quite obscure until the 1990s, when it was realized that a certain number of these immunoglobulin gene rearrangements go awry, particularly the class switch recombination. So instead of getting your looping out here, you get some other chromosome into the mix, so you get the immunoglobulin promoter enhancer linked up to some other gene. And this happens now in about 40% of cases of multiple myeloma, and there are 6 common translocations: cyclin D1, D3, CMAF, MAFB, and maybe it's 5. And one of the commonest is the MMSET rearrangement on chromosome 14. Chromosome 4, excuse me. So these are 414 translocations that drive the expression of MMSET as well as FGFR3. So this is because the promoter enhancer is linked 5' MMSET, driving MMSET, but at the same time there's a 3' enhancer that's linked downstream of FGFR3. So in many cases, these patients will overexpress both the MMSET gene and FGFR3. But in a certain minority of these cases, it's only MMSET. So MMSET universally overexpress FGFR3 most of the time, but not all the time. This would lead one to think that MMSET was the critical gene. So this just shows here the immunoglobulin promoter enhancer driving MMSET and the 3' enhancer driving FGFR3. Those are the 2 oncogenes that happen, putative oncogenes, that get rearranged and misexpressed in most cases, but not all. Every case has this. These patients don't do that well. These are older data, but they've held up over time that with bone marrow transplant, patients with the 414 translocation always seem to do worse. More recent therapies, such as the proteasome inhibitor bortezomib, seem to overcome this, at least initially. But we still worry about these patients that when they relapse, they're not going to be susceptible to high-dose chemotherapy, which can seem to salvage some people out to even very long time periods. None of these patients enjoy that kind of survival. So MMSET was really not known until it was cloned by this rearrangement. It really wasn't very much studied. And it's a multi-domain protein with notably a set domain named for funny Drosophila genes. SUVAR39 enhancer zest and trichlorox. And this is one of about 60 or 70 set domain containing proteins found in the human genome. Other notable ones such as Alex has worked on, thank you very much, include MLL, appreciate that. So set domain proteins are common recurrent motifs, and these encode histomethyltransferase activity, whereas these types of domains are likely chromatin readers, such as PhDs that Alex has worked on, PWWPs, which we'll talk about. So first thing we want to know about this overexpressed protein, is this really a driver phenomenon or some epiphenomenon? Is MMSET really important for the life of these tumor cells? So to do that, we did short hairpin. We made inducible short hairpin knockdowns of MMSET of 2 varieties. One that would eliminate all MMSET, a long form and a shorter isoform that only contains the N-terminal domains. And we found that in this type of knockdown, we got a change of behavior of these cells. They slowed in growth and they adhered to plastic. And this has been reproduced by others as well. So this affected biological activity, growth, and adhesion properties. And here we can see that there was some modest increase in G1 phase, and cells started to grow down over time. We did another knockdown that, this is an imaginary isoform. Some people think it exists. I don't think it does. But let's just think about this as just knocking down big MMSET, but not little MMSET, the shorter isoform. This led to apoptosis of these cells and pretty profound growth arrest and G1 arrest. So if we had something that would inhibit the enzymatic activity or just selectively remove this long MMSET isoform with the enzymatic activity, we might expect something like this to happen to a cell. So these types of experiments, and again, I said several other groups have done these as well, suggest that high-level MMSET is important for the life of these tumor cells. And we've transduced other types of myeloma cells without the MMSET rearrangement, and they don't seem to care so much about it. So these cells seem to be living on MMSET. So we think it's a validated target. We don't know how to target it yet, but we'd like to target it in this type of myeloma, which is, again, 15% of about 50,000 cases. So it's a sizable number of people per year, and it's not very well treated by normal chemotherapy. So after this, we'd like to ask whether or not biochemical analysis of this protein could lead to insights into disease pathogenesis and therapy. Just to review here, we think MMSET is a histone-modifying protein that will that will write certain marks onto the chromatin, the various sites that can be histone modified. And of course, histones can be methylated. I just learned today butynylated or butynylated, propionolated probably, but ubiquitinated and the like. So many chemical modifications, almost an astronomical number. And the set domains are histone lysine methyltransferases. So we'd like to know, does MMSET write certain marks on chromatin? And many chromatin writing enzymes also read the marks. They might read the same mark they write, they might read other marks or actually bind to them. So again, the histone lysine is one of the most flexible marks that can be modified in many different ways and recognized by different types of domains. And that's just a model of the set domain with an inhibitor actually locked into a groove here. So again, the probability of a gene being transcribed or not is going to depend, at least in part, upon what type of chromatin modifications are found on the histone tails. Activated genes would tend to have H3K4 methylation, and lack of methylation of these residues, whereas the repressed genes would tend to lose the H3K4 methylation, gain K27 methylation, and K9 methylation. So what marks does MMSET read, and how does it get to the nucleus in the first place? So what we found, one thing we found very early on when we were making reagents for MMSET, is that we would make this nice antibody, and then we'd make a immunoblot from cells, and we find no MMSET at all in the cell lysates. And we transfect it in, transfection's working great. What's going on? Well, it turned out that MMSET is very tightly chromatin bound. If you do some limpy kind of, low salt, low strength detergent assay, all the MMSET's in the pellet. So what we had to do is we actually do a chromatin preps to really treat the cells quite rigorously to get MMSET out of the nucleus. Except, and so these are 414 positive cell lines, 414 negative. These two cell lines have very long forms of MMSET. This cell line's got a shorter form because the chromosome rearrangement actually broke an intron exon boundary that eliminates this PWWP domain. So you make a shorter MMSET, which actually now appears in the nuclear fraction, not just in the chromatin fraction. So we would, this is still a cancer cell, and this still occurs in patients. So this domain may be dispensable for carcinogenic activity of this protein, but it might be very important for normal activity, as shown by its ability to normally be in the chromatin fraction, but come out into the soluble nuclear fraction and you lose it. And this correlated with immunofluorescence experiment, where MMSET was tightly bound to the nucleus and excluded from the nucleolus. But if we lost this PWWP domain, we start seeing it leaking into nucleolus as well. So this PWWP motif might be very important for tight binding of MMSET to chromatin. In fact, well, if you made one of these classical Dignum Raider nuclear extracts, we don't even find MMSet in those. So it might have been missed in some people's purifications because it's just too chromatin bound. We can get it out of the pellet if we add micrococcal nuclease to the pellets. Then you see here, it's all in the pellet. And now you start to see it coming out in the nuclear fraction. In fact, SYN3A is actually quite found in the pellet, some in the nuclear fraction. Virtually no MMSet is here, but if we add micrococcal nuclease, you start to see MMSet coming out. So Dinshaw Patel, who's a member of this score group, as is Dave Ellis, who Alex used to work with, crystallized this PWWP domain of MMSet, nice crystals. These guys know how to make proteins nicely, and found that it has a fold that resembles that of NSH6, suggesting that this is, he actually thinks that this is a DNA binding motif. Not particularly specific, but a DNA binding motif. And this is a work that we still have to follow up a bit more with Dinshaw on. So we think one thing, MMSet might be tightly bound to the nucleus and in part due to this PWWP DNA binding module. We have other experiments I won't show today showing that MMSet does bind DNA, but rather non-specifically. We don't think there's any sequence preference in it so far. What about those PhD in these other domains? Work, largely done initially, I think, by Joanna Winsaka with Dave Ellis and others in the lab, Alex amongst them, used a series of modified peptides that resemble histone tails. And we take these histone tails that have various modifications on it. They're bound to avidin Bs. We dip them into nuclear extracts expressing MMSet and ask which tails can pull out which portion of MMSet. And what we found was that MMSet binds to unmethylated histone tails. It doesn't like H3K4 methylated tails at all, and it seems to like to bind to H3K27 marks and K36 marks, seen over here. We found that this isoform here still retains some histone binding, but if we lose this PhD figure number 4, we basically lose everything. And so this protein still has the ability, we actually know it's still got some histone-methyltransferase activity, but it doesn't have very good chromatin binding activity. So we've actually been in touch with Dinshaw's lab, and we're going to start seeing if we can make some soluble PhD number 4 for some of the nice co-crystallization studies that he's done with Dave's group and Bob Rader's group. So we think that there are a number of very important modules here, but it looks like PhD finger number 4 might be in particular important. And actually, RDP1 has 4, but it's lost this PhD finger. It loses a lot of binding here. So the PhD finger that's lost over here, it still has this, but it's lost these others. These might be important, and these might be particularly important as well. But we're going to focus on number 4 in the coming year. Okay, so we think MMSet gets to chromatin because it has DNA binding activity, not specific, possibly mediated by PWWP domain, and it's got some histone mark binding, particularly likes to bind K36, which you'll see be important because that's the mark that it makes, and also seems to bind to K27, which is not the mark that it makes. So the question now is, what mark does MMSet make, and in the longer term, can MMSet inhibitors be found that might phenocopy the knockdown of MMSet in cell lines? So MMSet was really a pretty finicky enzyme. It took my graduate student a couple of years to get this to work. This enzyme always worked, first time, every time. G9A, it's a histone 3 lysine 9 methyltransferase. MMSet in vitro is promiscuous. It both methylated histone 3 and histone 4 in vitro by this radioactive label transfer assay. We incubate MMSet with radio-labeled S-phosphomethionine and naked recombinant histone. Now, we've got an even more active enzyme from Dinshaw Patel, and when we do that, we find the thing MMSet likes to methylate most is itself, at least. We're not sure if this occurs in vivo or not, but MMSet seems to methylate in a particular residue. We've mapped the residue, but we're not sure if it has functional significance. But you can find histone 3 and histone 4 methylated as well in this assay. And if we use antibodies against specific histone marks, we find again this promiscuity, not just on histone 4 and 3, but various histone 3 marks. In vitro, MMSet can methylate histone 3 lysine 36, 27, and histone 4 lysine 20. Danny Weinberg group also thinks it methylates histone for lysine 44. So in vitro, very promiscuous, and Danny Weinberg group had a paper suggesting that in different chromatin contexts, MMSET does different things. In fact, when we took nucleosomes, perhaps we got from Bob Rader's group, we didn't see any histone 4 activity, we saw histone 3 activity only. So, with this active enzyme in hand, we wondered whether or not we could develop an assay for MMSET inhibitors. And we used an assay based by Collazo and colleagues, which is based upon the depletion of S-adenosylmethionine from a reaction. So you need aldomet S-adenosylmethionine to cause this histone lysine methylation. If you deplete that, you get S-adenosylhomocysteine, which is then deaminate to form homocysteine with a sulfhydryl, which reduces thioglow, which causes a glowing compound. So, the more enzyme activity, the more S-adenylhomocysteine, the more glow. So this actually worked with our MMSET enzyme, but there is a background decay over time because we think the thioglow just sort of decays over time. This was the experimental and this was the corrected. And we could found, using the fluorescence assay, and we did this experiment is done at this plateau point. One drug called Pitocin, which was previously found to be an H3K9 methyltransferase inhibitor, also in a dose-dependent manner, this is no inhibitor and increasing doses inhibitor, inhibited MMSET activity. So there is an existing MMSET inhibitor. And this is a structure of Pitocin, it's actually another compound, BIX1294, bound into the GLP protein. This is BIX1294, another known G9 GLP inhibitor, and it also will inhibit, this is a not a radiolabel assay, but a methyl antibody assay. This is MMSET methylated histone 3 lysine 36, and with increasing BIX compound, you get less MMSET activity. Intriguingly, we have genetically matched cells with high MMSET and the same cell line knocked out for the rearranged MMSET. So they were matched cells otherwise. If you take the MMSET high cell and treat it with the BIX compound, you see this kind of death here. The matched MMSET low compound has less cell death. So this HMT inhibitor killed the MMSET positive cell line more readily. We're now going back and checking to see what kind of kinetics and what kind of effects we're seeing on histone methylation to be sure this is an on-target effect. But it's motivational to us to think that maybe you can find an MMSET inhibitor that might have a selective activity for an MMSET high myeloma cell. So to further this, and I see people from the Merck Research Lab here as well, we've worked with Milan's group using the SAMD assay, which he has pioneered, the solid state mass spectroscopy on a 384 well plate. So we've taken immobilized peptides, a very small peptide, and incubated, well, we first take the peptides, incubate with MMSET, immobilize it on these plates, and then put it through the mass spec machine. So we found that even a very small, basically 7 amino acid peptide could be methylated by MMSET. We could get monomethylation, and sometimes we can get dimethylation. I think Chris is here. Sometimes we get it with a good prep, we can get some dimethylation, I think. And then we put this through the, we could do 2 different types of experiments. One is to randomize amino acids alongside the central lysine with things being methylated, and ask what kind of specificity does the enzyme have as a completely naked enzyme in vitro. And the other thing we can do is we can use this high throughput machine to look for inhibitors of MMSET activity. So this is kind of complicated, but all this I want to take away from this is if you do indeed randomize amino acids alongside the central lysine that's methylated by MMSET, you do see some peptides some amino acids that give a lot of activity, and some amino acids that are just that give no activity whatsoever. So there is some specificity of this basic site and what MMSET will and won't do. whether or not that translates into in vivo specificity. Can we predict some methylation sites other than histones that MMSET might in fact methylate? That's where we'd like to go with this. Now the other thing to do here is we've taken the first 10,000 compounds that were done by Milan's group, and they were put into pools, and we then did a pool and split kind of strategy. And there were several candidates that came out here from the, I think it's the Cambridge Library. So I think 6 out of 10,000. I think 3 out of the 6 were out of stock, I believe, right? So we were able to get another 3 of those compounds, and 2 of them look like have validated in secondary lower throughput screens. What I've in fact done now is I took those compounds. I have a colleague who's a computational biologist at University of Maryland, Alex Mackerel, and he's gone, and I'll have to share this with you, we just got this. He's got a whole bunch of other structures that he would predict from this computer model that I showed before that might even be more interesting fits. So we're going to have a iterative process based upon what we come up from this and some structure-based, maybe rational design to try to come up with something that might be a good inhibitor. So we know that the enzyme is promiscuous in vitro, but what does it do in vivo, and what is its implications for living chromatin, not just these peptides in a tube? When we didn't really know that much about MMSET, we did what any transcription factorologist grew up in the 80s did, which was to tether your favorite transcription factor to the Gal4 DNA binding domain and see what happens if you tether to DNA with Gal4. Does it turn genes on? Does it turn genes off? What does it do? And so when we did that, we found that if we made a Gal4 of a little bit of MMSET, the N-terminal portion, or the full length, it tended to be a repressor. Compared to Gal4 1 to 147, here's some controls of Gal H-1. That's a pretty good repressor. But almost every domain seems to be repressive. In fact, we found that MMSET could co-precipitate with HDAC1 and HDAC2, as well as SYN3A and SIRT1, a whole bunch of co-repressors. And in fact, if we did this experiment, we took Gal HDAC1, that gave repression, and we added just free MMSET in the co-transfection, we got further repression of the Gal4 reporter template, suggesting that MMSET might be in some context a repressor protein that could nucleate or associate with SYN3A, HDACs1 and 2, and repressed genes. Now, why didn't you find it as part of the NCOR complex or the SYN3A complex? I think because you wouldn't find it because it's too tightly bound to chromatin. So unless you did this nucleosomal, this microcopyl nuclease freeing step, maybe you never found it. Maybe that's why it wasn't identified before. Or maybe it's just an occasional partner, it just hooks up every now and then, such as social life is nowadays. Anyway. So the other thing that we found is one of its occasional partners, or hookups, or whatever, was LSD1. And in fact, we found when we tethered MMSET to a Gal4 DNA binding domain containing reporter embedded in chromatin. No. Okay. Great. All right. Anyway. When you tether Gal4, this is MMSET's DNA, what we actually found is not only do we get on this reporter gene an H4K20 methylation, which in some contexts is a repressive mark, we also saw a loss of H3K4 dimethylation. And this corresponded with co-IP experiments showing that MMSET could actually bind to LSD1. So not only could MMSET bind to HDACs and cofactors like SYN3A, but it seemed to bind to a histone demethylase to, in certain contexts, and this is an artificial context, but at least in this context, it seems to have a repressive type of effect biochemically. So the model here would be MMSET, at least in certain contexts, could bind to a variety of these categories here. And we also found in a U2 hybrid screen, MMSET could bind to a number of Crab-Zig figure proteins. So MMSET might, through this putative complex, come and remove acetyl groups and add repressive methyl groups and shut down gene expression. So that's one model and that was our model going into the next set of experiments. We thought MMSET might be primarily a repressive protein. So when DNA, it can add a H4K20, remove H4K3, and deacetylates chromatin and represses genes, at least in that Gal4 context. But is that what it really does in cells? Well, the answer seemed to be somewhat different when we went back to our knockout and knockdown cell lines. When we knocked down MMSET by short hairpin RNA, we did immunoblots for every chromatin mark we could get antibodies for, we actually saw very little change, maybe even modest change in H4K20, very small, and not that convincing. But what we did see is when MMSET was down, we got a big loss in H3K36 methylation and an increase in H3K27. So this monomethylation is up because the di and trimethylation is down. And over here we see an increase in H3K27 trimethylation. This is the knockout, genetically paired knockout cell line. Same thing, K36 down, K27 here way up. If we take MMSET and we made several mutants that were based upon a model that Ming Zhao, a colleague back at Mount Sinai, did, he said if you, this over here would be, I think is the SAM binding, this is the histone binding area here. Make these mutants, let's see what it does to methylation. What we found is if we take these knockout cells and we add MMSET back into a cell, we gain K36 methylation. This mutant seems dead, it doesn't methylate at all. This mutant over here, right here in this pocket, seems to lead to more K36 methylation. And the reason for that is that this sterically unblocks the pocket so you can go from mono to di to trimethylation. This mutant has been made in other set domain proteins, and in fact this mutant is the natural type of mutant that occurs in lymphoma in the EZH2 protein, where EZH2 gets a mutation right at this phenylalanine residue. It goes to phenylalanine to, I think, a histidine in some cases. It causes less steric hindrance, and it turns EZH2 into primarily a trimethylation enzyme. So this type of AGAC experiment suggested to us in vivo that MMSET might be primarily doing this K36 methylation. And this cell line experiment, it kind of looks like gibberish, but it's easier when you look at it like this. We take a look at a collection of every existing 414 cell line we could get our hands on. They have somewhat different size MMSETs because, as I said, the chromosomal breakpoint can be a little different. But it's all much higher than over here. In every one of these cell lines, we see an increase in H3K36, primarily dimethylation. The trimethylation looks pretty even. The trimethylation looks pretty even, actually, but the dimethylation is quite distinct. And the K27 methylation is way down over here. So there's a global switch. I mean, when you see this by immunoblot, this means that millions and millions of nucleosomes have had a very significant change to their chromatin structure. And with Neil Kelleher, who was at Champaign-Urbana, and is now on the Evanston campus, we did a mass spec analysis using his so-called top-down approach, where he could look at large, instead of fragmenting the nucleosomes into little bits, he takes really big protein fragments and put them through very strong magnets and can measure large ions. And what he found was that, particularly histone 3.1 and 3.2, we had an excess in these MMSET high cell lines of these peaks over here, indicating there was increased molecular weight of these histone peaks. Seen over here as well. Not on histone 3.3, interestingly. And what we found is that when you have MMSET high cells, if you count the ions that have histone 3 lysine 36 dimethylation, it's like 70% of the ions have that dimethyl mark. In an MMSET low cell, in fact, we realized that there's been a lot of work done on histone 3 lysine 36 trimethylation, but this is a non-MMSET expressing cell shown here in these white bars. And here's the dimethylation, here's the monomethylation. Actually, K36 trimethylation is a very small fraction of a normal cell. It's really dimethylation is more where the action is. And in MMSET high cells, there's a whole lot of H3K36 dimethylation. At the same time, MMSET high cells have very low levels of H3K36 trimethylation, and a lot higher levels of dimethylation and monomethylation K27. Is this important biologically? When we took MMSET with a point mutant, this mutant that was absolutely dead for HMT activity, we added it back to the MMSET deficient cell. The dead mutant didn't do much difference compared to the MMSET knockout cell. If we put MMSET wild type or the hyperactive mutant back into the cells, we got accelerated growth. So this suggests that the H3K36 methylation activity of MMSET is one critical component of its ability to stimulate cell growth. This dead mutant, not very much. This live mutant could stimulate growth more. So we finally call this HMT's gone wild. That the MMSET is just causing a global epigenetic change because it is at present high levels, and it's modulating the chemical structure of millions of nucleosomes throughout the cell. The K36 is high and the K27 is low. So how does this correlate with gene expression? What does this do? Or is every gene expressed differently, or only some genes? Well, one bottom line is it's only a few percentage of the genes in the cell seem to be significantly affected, maybe 800 out of 20,000 assay. And that's in some ways not so surprising if you look at studies where people have taken histone deacetylase inhibitors, which globally change chromatin, and ask what percentage of genes change. 3 to 10% is the range. Same kind of range here. So global chromatin change, but only a limited genetic expression output. And again, we use these mutants to actually, this point mutant that's dead in activity as one important assay to look at what MMSET's activity is on average on the cell. And the way I did this, and this is my own bioinformatic activity using Excel. Nothing more than that. If you plot all the gene expression changes, okay, the gene expression changes came out of GeneString, but if you just made a list of them and just said, order them from highest to lowest in a so-called waterfall plot, you look at the genes regulated by MMSET alone, you got a lot of genes that are like upregulated about 60 to 80 fold. And the 90th percentile of these was 4.5 fold, and the 95th percentile was probably 50 fold or something. These are regulated by MMSET wild type with HMT activity. MMSET regulated genes with the form of MMSET that's the HMT is inactive. Still some genes are turned on and off, but they're only turned on and off about 2-fold. We see none of these genes at the tail way up there at the 80-90 fold. So to me, this would suggest, in this low-tech bioinformatic approach, that MMSET, and in fact, when you have genes that are regulated by both the mutant and the wild type, it seems that when those genes were affected this way, a gene that would be affected, let's say, 2-fold by the HMT dead mutant would be upregulated like as 5-fold by the HMT wild type, suggesting that MMSET has an activation mechanism as well as repression, and the HMT activity is critical to get very high upregulation gene expression. So to put that together, this is just inferential, but says that MMSET, yes, when tethered to DNA, might nucleate repressive complexes, but when overexpressed in these myeloma cells in this global way, seems to have a generalized effect to upregulate more genes than it downregulates. And that's HMT dependent. And this could be a tumor-specific effect. It may not be related to what MMSET does normally, because it's not normally expressed at these very high levels. Again, with the caveat that this may be a tumor-related effect, it may not be related to normal MMSET function. We ask what genes are regulated in response to these high levels of MMSET. And we can do this by doing knockdowns followed by microarrays or add-backs of MMSET and looking for genes that are turned on only by the active mutant. And there's some genes that are turned on by the active and the dead, and some only by the active mutant. Well, anyway, when we do this, so we look at the genes that are regulated by MMSET, we tend to get genes involved in apoptosis, DNA repair, which we'll see will be interesting in a moment, and the cell cycle control. And that's not that surprising. Almost, many oncogenes will do things like this. What we don't yet know is which genes we're working on, what genes are directly bound by MMSET, and direct binding is critical for their gene expression, and what genes are just regulated because their chromatin's changed, maybe not even by direct binding, but they're just susceptible to be changed. And that type of analysis, I think we're going to have to do some important promoter motif analysis, and maybe there's a hallmark of a gene that's regulated by MMSET, but not bound per se, and other genes that are regulated because their promoters are in such a configuration that if they get that chromatin change, they will actually be changed in expression. Here's one example of a gene regulated by MMSET and activated by MMSET. JAM2, which is an adhesion-related gene, is not, when you knock down MMSET, it goes down. In the knockdown or knockout cells. And we add back wild type, but not dead mutant MMSET, the expression goes up. Now, in this gene, we did do chip assays across the locus, and we found in MMSET high cells, the entire gene seemed to be coded with MMSET. And maybe this is just because it's a sticky protein and it's expressed highly in the cell, it just gloms on everywhere. We don't know. But it's off at higher levels in MMSET high cells as opposed to genetically matched MMSET low cells, and that correlates with higher dimethylation and trimethylation across the entire gene. The MMSET high cell tends to have less K27 methylation in the promoter, not in the gene body, than in the MMSET low cell. There's higher K27 methylation. Now, more recently, we've gone from identifying potential target genes by differential gene expression to doing ChIP-seq approaches. And what we've seen here is that we found it was a little tricky. We had to use a double cross-linking method. We used ChIP-seeker software. We found that MMSET tended to bind mostly in introns, mostly in first introns. So when we were using chip-on-chip ancient history of 3 years ago, we would have missed most of the action of where MMSET might be binding in the genome. Again, very few of the total sites were promoted. Real enrichment in introns, and particularly first introns. And here's one interesting link we found, one gene that came up in our differential expression list and bound by MMSET, this DNA methyltransferase 3B. This is a K36 dimethyl tract showing that MMSET high cells, there's a lot of K36 dimethylation throughout this gene as opposed to MMSET low. And this correlates with high MMSET expression here and the high K36 over here. So this is interesting because we already have a little preliminary data that there's a dramatic change in DNA methylation in these MMSET high cells. So there may be a link, maybe one link is that MMSET actually changes expression of DNA methyltransferase 3B. So MMSET target genes so far, we think some are set domain dependent, they require that HMT activity, and some are independent. And which of these gene pathways are most critical for pathogenesis is something we're still trying to determine. So if MMSET has changed chromatin so dramatically, is it all about gene expression, or could there be other problems with these cells? And as part of our PSOC, Michel Lebeau's group is actually looking at the timing of DNA replication in different replication origins in our MMSET high and low cells, and there are distinct differences. So one possibility is that global chromatin change might change something about the order of normal DNA replication and perhaps the fidelity of such processes. But one thing we want to do is we want to see, did this big covalent chemical change lead to a change in the physical properties of chromatin? So what we did here is a micrococcal nuclease accessibility assay. We treated bare nuclei of MMSET high cells with micrococcal nucleus, so we get this laddering effect. We knock down MMSET, we lose that laddering effect. It's like the chromatin scrunched up, it's no longer accessible to micrococcal nucleus. If we reverse the knockdown and turn MMSET back on, you gain the laddering back on. So it's like MMSET high cells seem to have high K36 methylation and a fairly loose chromatin accessible to micrococcal nucleus. What other process might, when I was thinking about this, saying, well, what other process might be regulated differently with different physical state of chromatin? And one thing that came to mind was DNA damage. So in fact, we found that these MMSET high myeloma cells have a basal activation of DNA damage pathways, as shown by staining with gamma-2AX. And the knockout, genetically matched knockout cell has much lower staining. If we add radiation, 5 or 10 gray radiation, we get more H2AX foci here than over there. But there's a bit of a paradox here. So the MMSET high cells have more DNA damage. What if we treat these cells with the mainstay chemotherapy drug for myeloma until the past 10 years, a drug called melphalan? What we found is that the MMSET high cells seem to survive treatment with melphalan better than the MMSET low cells, the black versus the white. And I think I took the one slide. Let me just change my thing. And when we did with these are common assays, single cell electrophoresis assays, MMSET, these are control cells, and we added MMSET to these cells. It looks like we got elongated comments. We put the HMT dead mutant in here, which wouldn't cause all that K36 hypermethylation. We didn't get those comments. And over here, if we combine MMSET and gamma radiation, we got very long comments, and less so if we added the HMT dead mutants. Furthermore, if we took MMSET high cells and we cultured them for a prolonged period of time with melphalan, we found that MMSET high cells failed to go into cell cycle arrest. There's still cells here that are BRG positive. MMSET low cells went into cell cycle arrest. So this is kind of bad. You're getting DNA damage, but you're not going into cell cycle arrest, and you're surviving better. This is not a good thing for your tumor cell. You want chemotherapy to kill a cell, cause DNA damage, and let it die. Here it looks like they're getting more DNA damage, but they're not dying. So this is not a good thing. Okay. Let's go back to you. So finally, I just want to mention some things we've been trying to do with your center in the ISGB. One thing we recently received a grant from the Myeloma Foundation to see, can we find some other kinds of Achilles heels of MMSET-associated myeloma? And what we did is we curated it. We said, what is special about MMSET high myeloma? We took patient data sets and we looked for what are the genes that are consistently overexpressed in these patients. And we collated, and we didn't really do a Venn diagram such as to, we did a union instead of the union of the sets rather than the what's only found in 2 sets. And we found about 344 genes that seem to, in one data set or another, seem to be upregulated in patients with MMSET high myeloma. And, okay? Anyway, I'll meet with you in a few minutes, okay?
You're on.
Okay, thank you. So these 400 genes or so might be genes that maybe they're highly expressed in MMSET high myeloma. So our idea was to knock them down using siRNA in these MMSET high cells. This is a positive control, which is IRF4, shown by Lou Stout to be critical for, we can knock down MMSET and we can lose some K36 methylation. If we knock down IRF4, we lost cell viability. So that's kind of where we're at right now. We're still working out some kinks with the screening center. But our idea is to say MMSET high myeloma has a certain gene expression set. Some of those genes might be direct MMSET targets, some of them might not be. But these 400 genes might be represent unique pathways or genetic targets that might be targeted in that form of myeloma. And the other thing we've done with the center here is we've taken the Prestwick compound library of 1,000 FDA-approved compounds, and we asked just what kills an MMSET high but not an MMSET low cell line. And we got a number of hits here, things that will differentially kill in the MMSET high but not the MMSET low cell line. And we've gone back and rescreened those, and we have about, I'd say about 24 compounds, things like antihypertensives and diabetes drugs that we wouldn't have thought of could be repurposed in this way, but might be, potentially leads to treat MMSET high associated myeloma. So I'd like to round this out with a few conclusions. We think that MMSET rearranged in a fraction of patients with poor prognosis myeloma is a therapeutic target. If we could get rid of it, that would be a good thing to get rid of the myeloma. That there are MMSET inhibitors, some of the drugs, BIX1294 and Pitocin can actually inhibit MMSET activity, but they have rather broad activity. So the question is, can we find a specific MMSET inhibitor, and could that ever be a therapeutic agent? MMSET can form repressive complexes, repress genes, and it can also activate genes through its global histomethylation, and it may have effects on other chromatin-dependent processes, such as DNA repair, and as I said, Michelle's lab makes some information on DNA replication. And we also think that, like many tumors, the gene expression network set up by MMSET, because of its peculiar properties, might set up specific Achilles heels. It might be that cell is susceptible to knock down certain genetic pathways or to certain drugs. And just in the last moment, is this only relevant for multiple myeloma? Well, maybe not. Curating databases like Oncomine, you can find that MMSET is highly expressed in diseases like metastatic prostate cancer, along with EZH2. And it does the exact opposite of EZH2. EZH2 makes K27 marks, MMSET makes K36 marks. And those are kind of antithetical marks. They really don't exist together. If we knock down MMSET, at least in 2 out of 3 cell lines we tested, we found that knocking down MMSET prostate cancer cell lines inhibited cell growth and inhibited colony formation soft agar. And this type of work has been recently published by Kristen Hyland for neuroblastoma, that high levels of MMSET seem to be an Achilles heel of neuroblastoma. If you knock down MMSET, the neuroblastoma cells would stop growing. So if we had MMSET inhibitors right now, they might have broader implications than just this one particular genetic subset of multiple myeloma. So this work was done by my group at Northwestern with collaborators including Chasey Leaf-Bergsig, who cloned MMSET, Lou Stout, who developed a lot of these knockdown cell lines, Dinshaw Patel, as I said, the group, initially Zach and more recently Chris from the University of Chicago. And here are some of our funding agencies, including our SCORE Consortia and all these other agencies. And I really appreciate the invitation, Kevin, and the chance to meet with some of you here today. Thank you.
Okay, so we'll take a few questions. Maybe I'll get started. So I guess I have a question about...
And records. And this red light.